Avidity is also working on other exons in DMD. We're working on exon 44. We're working, in addition, on exon 51. And the goal here is to be able to have drugs that will be suitable, therapeutic agents that will be suitable for skipping multiple or different exons that will serve a larger population of the boys that suffer from this terrible disease. And so we're moving those forward as well. Those are following behind exon 44. But what we're doing in general is developing this concept that you can take advantage of the transferrin receptor to deliver these important therapeutic agents into the cells of muscle. And that includes cardiac muscle as well as potentially smooth muscle. So the platform that we're developing around muscle is ideally suited to a number of different neuromuscular diseases. So we're working on DMD, as you heard, exons 44, 45, 51, and potentially others. But in addition, we're also working on programs in myotonic dystrophy. So myotonic dystrophy, rather than being a disease that's caused by a out-of-frame mutation in a particular gene, myotonic dystrophy is caused by a toxic messenger RNA, where the messenger RNA for a particular gene known as DMPK has a number of CUG repeats in it. And the excess number of CUG repeats leads to a condition where those CUG repeats accumulate in the nucleus of cells, and then inappropriately bound to those CUG repeats is a protein called muscleblind-like protein. Muscleblind-like protein is a critical protein that's used in splicing and the processing of other messenger RNAs to make other proteins. So if all of the muscleblind-like protein is inappropriately sequestered against binding to these overexpressed CUGs, it's not going to perform its downstream function. So the phenotype, the disease associated with myotonic dystrophy, is associated with a loss of function of MBNL, muscleblind-like protein, and a missplicing or misprocessing of a number of different proteins that are critical for muscle function. So our first drug is in the clinic, AOC 1001, that's treating myotonic dystrophy. And we talked at AAN about the clinical trial design for that particular clinical trial, a phase 1, phase 2 trial in patients suffering from myotonic dystrophy type 1. And so that program is moving forward. And again, we're looking forward to talking about some of our interim results later this year, in the fourth quarter of this year, which will of course be extraordinarily exciting. Because at that point in time, that will be the first proof of concept in humans that we can utilize a monoclonal antibody to the transferrin receptor to deliver these important therapeutic agents into the skeletal muscle of patients suffering from neuromuscular diseases. And so that's an amazing realization of an amazing dream that we had just a few years ago, that we could utilize this mechanism in order to deliver oligonucleotide therapeutics to muscle. And being able to demonstrate that in a human clinical trial, of course, is the first step. The next step is, of course, to demonstrate that you get reduction in the target messenger RNA. We want to reduce the amount of that toxic DMPK messenger RNA. So that's also an important proof of concept. And then, of course, we have the patients. And hopefully, as a result of treatment, we'll see improvements in various parameters associated with muscle function in those patients. All very early in a relatively small phase 1, phase 2 study. But again, the proof of concept for the technology is so important to those patients who are suffering from neuromuscular diseases that can be addressed with oligonucleotide therapeutics.